Okay, thank you so much for introduction. I really like your pronunciation of my name. It's usually a big task for people. So yeah, my name is Ilja, presenting you from University of Notre Dame, and I'm going to talk about infrared photothermal imaging, how we apply it to material research. I also touch on what we think the origin of signal in this technology, in this technique, and in the end, I will briefly talk about applications in environmental sciences. So just to give some historical overview, where it's all started with a single molecule and single particle measurement. It all started in 1989 with William Merner measuring absorption of single molecule using very complicated techniques like double modulation. This was followed by Michel Arid, who's present in this webinar, who detected single molecule fluorescence just a little bit later. And arguably, these are two works that kickstarted the whole research field of single molecule spectroscopy and let's say single particle spectroscopy. And this has really helped to comprehend fundamental physics and chemical processes on a level beyond averaging on a single particle level. And people really like fluorescence measurement because they're easy, they're zero background experiments. You kind of can just detect it, emission on a black background. But fluorescence measurement have its own drawbacks, and first and foremost, to measure fluorescence, your sample needs to be emissive. And from electronic structure point of view, fluorescence gives you information only about lowest excited states and leaves out all other states unknown. And in search for robust absorption experiment, next breakthrough actually came again from Michel Arid lab with the photothermal measurement, which was then, let's say, perfected by Brahim Lunis later. And technique calls visible photothermal heterodyne imaging, or just PHI. And the technique uses photothermal approach to detect absorption of nano objects embedded in some photothermal media. And ideal case study here, ideal sample are plasmonic nanoparticles. They have very large absorption cross sections. They release a lot of heat. They absorb a lot of light. And so the technique is basically uses modulated heating beams that is on resonant with a sample absorption. And when that is focused on a sample, when sample absorbs this radiation, it releases a little bit of heat into medium, and this creates a thermal lens effect. And if you remember Michel Arid's talks and Frank Stitcher's, they really describe it well. And this thermal lensing effect is being probed by a second of resonant laser beam in an invisible PHI. It was a redder wavelength, so its resolution was worse. So what we're interested in, and what's actually was is infrared imaging, is infrared region of the spectrum. And the reason behind it is because infrared light gives you chemical specificity, and imaging is quite easy and fast. And FTIR microscopy and FTIR spectroscopy is well-established analytical area, so it's well understood. Problem with FTIR microscope with infrared imaging is that first you have a very long wavelength, and that means that your resolution also limited by this wavelength. And for FTIR measurements, for example, you're limited by very low sensitivities. And here is just a typical example of FTIR microscope, that very large field of use, very fast measurement, but very poor resolution that is limited by the wavelength of light used. Of course, they were developing how to break this resolution issue, and it started all in late 90s with coupling infrared radiation to near-field optical techniques and coupling it to AFMIR measurements. to AFM tips. And this technology gives you very high sensitivity with extremely high resolution. Now it's down to 6 nanometers, I believe. But arguably, this technology is very complex to operate. They require complicated sample preparation and gives you very slow measurements over very small field of view. Another technique even more extreme, this used for material science mostly, but so steam EELS, it can give you actually access to whole electromagnetic spectrum in one measurement with some unthinkable spatial resolution under 1 nanometer, but it's not instrumentation that you readily find in your lab or in your university. And additionally, this measurement performed under high vacuum with extremely monochromatic electron beam. And so there is no way you're going to measure water sample or biological sample. So now, combining this all together, there is a way to achieve super resolution with infrared measurement and to increase sensitivity of, relative to FTIR microscope by combining this all together. And we call the technique infrared photothermal heterodyne imaging, but you also might see some alternative names if you like OPTIR or MIP if you've been here last Wednesday on Professor Cheng's talk. And there is a commercial unit based on this technology called Mirage. An approach here is similar to visible photothermal imaging, to use pump tunable laser. In our case, it's just OPO or QCL that is focused through a reflective objective on a sample. Sample, if absorbs infrared light, undergoes non-radiative recombination, and as a result of this recombination, it changes its temperature a little bit, temperature of sample itself and surrounding media, if there is a media. And as a result of temperature change, sample experience thermal expansion, basically expands a little bit. It changes its optical properties. And these optical properties change are sensed with a second of resonant beam, here depicted as green. And the beauty here is because we use a visible probe beam to detect absorption of infrared radiation, we get infrared absorption data on visible light spatial resolution. We use so-called counterpropagating geometry just to take advantage of the best possible objective to focus probe laser and get the best possible resolution. Another advantage here is that because this is all optical technique, we have ability to get to extract other optical information such as visible emission, or we can complement it with Raman measurements, for example. Again, just a quick historical overview. Technique was first introduced in 2009, but never met, it didn't become very popular at the time because there was no super resolution demonstrated, just demonstrated chemical specificity that you can actually measure this photothermal. You can measure chemistry using photothermal methods. This was later in 2012, where again several American works showing that you can get very nice spectral fidelity and increased sensitivity, but no super resolution. But technique became really popular in around 2016, 2017 era, when Ji-Shi Cheng and Greg Hartland from Notre Dame introduced their technologies, their way to get it, and they also demonstrated that sensitivity is extremely high. It's down to 10 micromolar, better than Raman. And the resolution also is here, 600 nanometer from Professor Chen group, and 300 nanometer resolution using counterpropagating geometry from Greg Hartland's lab. Just again to keep you up to date with the technology, if you've been here on last week, you remember Professor Cheng's talk that they also now introducing this wide field geometry to also get around raster scanning images, which basically hinders speed of the technique. And this is just an example of wide field illumination of pattern, and showing that you can detect this photothermal signal using a wide field geometry. So well, let's come back to us, and this is the current state of IRPHI in our lab. And here I'm just showing you some of the better images that we've been able to take. And on the left are 100 nanometer polystyrene particles, image at the different wave numbers. Is a very reasonable signal to noise, very distinguishable. On the right are 300 nanometer PMMA particles. Not that we couldn't measure something smaller, rather that that's what I could buy at the moment. And again, now after Brassin scanning sample, after allocating areas of interest, particles of interest, we can take a local infrared spectra, a single particle infrared spectra, by recording IRPHI signal as a function of wave number or as a function of different lasers. And spectral fidelity for polystyrene, by comparing it to FTIR, and for PMMA is looking very good. So technique works as advertised: super resolution imaging with great spectral fidelity and very high sensitivity. So question that we had is what is causing this signal to arise? And the reason we were asking ourselves is because for infrared measurements, we were avoiding using medium. So there is no photothermal medium around sample, and there are several reasons behind it. Well, the biggest one is we want to avoid any competing on interfering infrared absorption with the sample. And so it's what I'm showing you as some earlier numerical simulation from Greg Hartland's lab. I will just briefly touch it. And here, so numerical simulation shows that absorption of infrared radiation did localize within the particle, and all changes are localized inside the particle. So there is no contribution from substrate or no contribution from air, which is basically surrounding medium here. And this absorption causes two effects to happen: thermal expansion, which increases scattering cross-section, and as a consequence of thermal expansion, refractive index decreases a little bit because of thermal optic coefficient. And these effects actually counteract each other. This creates a complicated signal dependency on size, and we decided to expand it with analytical modeling. And with analytical modeling, we actually confirms and actually expands this numerical simulation. But so for analytical modeling, photothermal changes or signal contrast as a function of changes to backscattering cross-section given our collection and backscattering light. And what we can do here is explicitly account for changes induced by thermal expansion and changes induced by thermal optic coefficient in the response to temperature change. And temperature changes just absorption of infrared radiation. And comparing it to real experiment, so here for different samples with different sizes, we see that it agrees very well, and it captures a very steep signal drop for very small particles when it starts when it hits a relay scattering regime. So this is very exciting for us. But what was more important for us, or equally important for us, is that we can see what are relative contributions of these photothermal changes to backscattering cross sections, again in response to change in refractive index, thermal optical effect, and change in physical size or thermal expansion. And what we find is that for larger sizes, at least for samples that we study for this polymeric samples, it's thermal optical coefficient contribution to backscattering intersection always dominate. However, it's not always true, and for certain cases, you can see that it's flipping, so it's very complicated signal geometry or signal dependency when you have to measure different sizes. As I mentioned, there is no surrounding medium in our experiment. That's what differs from main difference from visible PHI. Surrounding medium, however, can influence the signal, and this is because it's going to influence refractive index contracts most of all, and it's going to change how heat dissipates. It's going to change how thermal optical coefficient change, and basically it's going to influence how much changes to backscattering intersection is going to happen in response to temperature change. And results actually quite interesting here is that, so analytical, at least our modeling shows that if we embed some samples in a medium, if we increase in medium and conduct experiments on larger sizes object, it actually will decrease the signal, so it will hurt PHI signal contrast. However, when operating approximately below half the probe wavelength, effect is opposite and signal increases. And what's good about it is because for these conditions, when you have a dipole emitting heat in the medium, there is a well-established theories, and like it's all well understood in this region. And our suggestion is basically, if we want to move further into nanoscience with these measurements, we will have to embed a medium for our measurements. So now I'll just touch material science that we already did with these measurements. And first, I will introduce you systems that we study, and this system calls metal halide perovskites. You perhaps heard about it, maybe not, but in general, stoichiometry of this materials is ABX3 with it's a mixture of ions with on A side there's methyl ammonium organic cation from imidinium or cesium. On B, you always gonna see lead, and X it can be a mixture of chlorine, bromine, and iodine, but in our case it mostly will be iodine. And this is not very new materials. They've been discovered more than 100 years ago, but they became really popular in the last decade. And what I'm showing here is their great bull run is a solar cells, and their record power conversion efficiency for solar cells is now higher than polycrystalline silicons. But what's remarkable is that these materials took just less than 10 years to reach these heights. For silicon, it was 50 years of refinement. So, but because this is material started to be very popular very lately in the last decade, and there is sufficient body of research need to be done, and especially on microscopic level, because it's going to help to improve solar cells in the future. And what we're going to study is chemical distribution or chemical homogeneity, and how it affects solar cell performance or optical properties of material. And first test system is a mixture of organic cations in lead iodide in lead iodide perovskite. So it's 10% fermimidinium, 90% methyl ammonium. And what we're going to monitor is CN stretch for fermimidinium at 1710 wave numbers, and methyl ammonium band, NH band at 1465 wave numbers. And what I'm showing here is just chemical distributions over 10 by 10 over microns, so very microscopical area of methyl ammonium, and same area chemical distributions of fermimidinium. And by looking at these two maps, there's no exactly difference or very different but differences are really picked up when we take a ratio map of these two, simply dividing map A by map B and getting that's map C or ratio map. And now we can see that yes, indeed, there's a clear chemical heterogeneity exists. There are some regions in the films that are formamidinium rich, some regions in the films that are methylammonium rich, and this further can be corroborated by local infrared spectroscopy and by looking on relative strengths of as formamidinium vibrational transitions at this location. We can say that yes, there is different ratios of methylammonium to cesium. So this is a first observation of this phenomena. And what I didn't really mention you is that the technique itself reports you almost directly on infrared absorption, but it doesn't tell you exactly how much there was infrared absorption. You can do it in a relative way. So, but it's not quantitative as it on its own. And the way around it, like at least the way we saw, is to calibrate our signal for particular sample. And the way we did it, we just recorded again this signals of IR-PHI microscope as a function of chemical composition X in this chemical formula, and by simply calibrating signals, creating calibration plot. And now I'm showing you a second test system, 20% formamidinium, 80% methylammonium. And now I'm creating this ratio map as I showed on the previous slide. And now by applying this calibration plot, I actually can extract real chemical stoichiometry on local microscopic area. And as expected, it's average at 0.2 as an average, but stoichiometry changes up to 50% exist in local, and this is something really concerning. Yeah, and beyond this, we can also have access to optical measurements, and in our case, we extract visible light emission information for the luminescence. And now I'm showing you third test system, 30% from medium 70 methylammonium, so it works for everything. And so again, by creating this local chemical stoichiometry, chemical composition map of this system by the way I just show you, and by recording photoluminescence from the same area, from the same spatial location, we can now compare how the chemical stoichiometry translates into photoluminescence properties. And specifically what we're interested in is the photoluminescence energy, as it's gonna give you an idea what's happening with the band gap of these materials. And by correlating these two maps, we see that there is a strong correlation between local chemical composition and local photoluminescence energy, indicating that yes, indeed, this chemical stoichiometry translates into changes in optical properties. And the reason this is important from solar cell perspective is changes and differences in band gap. Band gap variation is going to translate into differences in open circuit voltages. And that's going to mean that your power conversion efficiencies will be different locally, as it's going to bottleneck performance of such solar cell. Yeah, beyond this, we actually can also monitor dynamics of this organic cation. This is next work. I will just briefly touch it. There's too much material science inside, but IR-PHI was very instrumental to establish organic cation migration inside working solar cells, inside working devices. And to do that, we study this perovskite thin films deposited between two ITO electrodes with a 40 micrometer channel, so we can apply voltage and mimic solar cell performance, basically imitate real operational conditions. And by taking ratio of, then we can apply in situ voltage on this microscope and image same areas and look for changes in distribution of organic cations in this case. And by taking again ratio maps from after applying voltage versus before applying voltage, we actually confirm that cations do indeed migrate inside solar cells. This is map showing depletion of cation from positive electrodes and accumulation at negative electrode. It's very well seen on a ratio profile. Here is just each point is a average of each line, and basically showing depletion from one electrode and accumulation in another. Same is true in more complicated systems like formamidinium, methylammonium, cesium mixtures, and both formamidinium and methylammonium do deplete from one electrode and accumulate at another. And this is really important. This is really disturbing because crystal structure is very defined, and when you have depletion of one of the ions, you actually degrade this material, you degrade your solar cells. So last, very quickly, I'm going to touch about something not scientifically challenging, I'd say, but very important topic, especially at least very important topic for me, is plastic contamination, is a plastic environmental contamination. And one of the areas is contamination by micro and nanoplastics. And this has been really had discussions in the last 5-10 years, and people been showing that this microplastics can and nanoplastics can come as a direct release from consumer products. And here is an example from 2019. So there is a individual tea bag, plastic tea bag that you can buy at the store, and instead of tea, I mean, we see it also releases billions of microplastics in your drink, and this is a concern. But beyond this, it's also been shown that polypropylene bottles for baby formula feeding also release lots and lots of this microplastics. So people really concerned. But one problem exists with characterization is how to characterize them in one shot. So you want to get your chemical information, you want to get your history of particles, you want to count them, and basically you need to single measurement to do so. Best bet here is FTIR that can give you both chemical information and particle counting, but as you remember, FTIR limited to particles around 5 to 10 micrometers. And SAM, on the other hand, is like perfect for counting, but zero chemical information. So what we decided to use is use our instrument IRPHI to study this microplastics, as it gives you chemical information, A, and B, it has better spatial resolutions than FTIR or QCL measurements. And here just we decided to reproduce or repeat and see for ourselves that actually making tea with a plastic tea bag is a better idea. So we just steep a single tea bag in a cup of water that's similar to what we usually do with tea, and then drop the suspension onto our substrate, onto calcium fluoride, and image particles with in resonance with a nylon CO stretch. And basically, yeah, we see there's a lot of particles present there, different sizes from several hundred nanometers to a couple microns. We can further take single particle infrared spectra to confirm that indeed this is all nylon, this is all released from tea bag. And by taking lots and lots of these measurements, we can come up with a concentration, like how much particles was released into the water. And we can compare it to SM, of course, and from IRPHI measurements, we get numbers that, to be honest, 10 times less than reference that I show you, but it nonetheless a very high number, almost just shy under 1 billion particles per cup of tea. From SM we get number 3 times higher, and this is quite a discrepancy. But the reason for this discrepancy is because SAM should be considered an upper limit counting because it has high number of false positives, and because there is no chemical information exists, just counting. For IRPHI, however, it's opposite. You get 100% zero false positives, but what you get is limitation in spatial resolution, limitations and detection limits, that you getting lower end numbers or lower limit. Beyond this, I mean, it's not just something simple like tea bags. It's actually, as microplastics exist everywhere. And here's just a couple of examples of general household items. So this is a dust from a household tobacco. And indeed, there's lots of particles present, a lot of plastic. Most of these particles are coming from nylon. There's what carpets are made of. But what's interesting, some smaller particles show features only correlated to nylon, while larger agglomerates have some extra features that can come from, for example, PET plastic, or that's what water bottles made of, or maybe PMMA or something else. But this is concerned. This is what you inhale in your apartment, your home. Same is true for, this is very relevant for America, for lint filter for clothes dryer. Not sure if people use it in Europe, but basically, again, this is something that is in your apartment all the time that you run weekly. And again, there's lots of fibers present, lots of fibers from polyamide, as it's now coming probably from some sport clothes and so on. But also there are features that are and locations that are really high and show very good correlation with ABS plastics that is again universal, present in your apartments, even legal made out of it. And this they are again create complicated structures together, and this what's shown here is that basically by imaging at 2 different wave numbers, we can see that like this area is, for example, this position has a lot of polyamide fibers, but when imaging and resonance with ABS, you see that distribution changed. For example, this point completely disappears, so there was just polyamide fibers, but other areas actually growing relative signal, so there's more ABS plastics than fibers. And with that, I just like to briefly mention other applications that is possible for this technology, and I want to point on plasmonic nanostructures. That's something that Greg Hartland is going to talk about later in this webinar series, so you don't want to miss it. And I want to thank my collaborators, my advisors, and everyone who was helping me. Ken Kuna, my advisor, Greg Hartland, also present here. Pavel, who was helping with all analytical modeling, and Kyle, who's our environmental engineering supporting person. And also I'd like to thank National Science Foundation for support of this work, but I also should disclose that views in this presentation are my views and not National Science Foundation. And thank you for your attention.
Thank you very much, Ilja, for the nice overview of what you can do in the field of material science with the infrared photothermal imaging technique. I'm pretty sure that there will be some questions. Either directly speak up, ask the question, or type your question to the chat, and I can post that. So Michel has a question.
Yeah, thank you very much. A very nice talk. I was surprised to see that in your simulation of the heating of a particle, the infrared actually is absorbed at the beginning of the particle. So what is the penetration depth of infrared? Of course, it depends on which band, but typically I thought it was rather weak bands. Can you comment a little bit on that and tell us how far the radiation penetrates into these materials?
Yeah, so in general, like our simulations were all on sizes below infrared wavelength of light, and so, like under 1 micron, and we assume that penetration is 100% there. But yeah, if it's even bigger, if it's beyond 10 micrometers, then you meet this exponential decay of absorption. And
okay, so in this case, it's several micrometer.
Yeah, yeah. So it's
okay. Yeah. All right, then. Thank you.
Okay. More questions?
Yes. I have a question. Okay. How easily can you take advantage of temperature as a variable? You see, it might be that, well, if you decrease temperature, then yeah, the contrast which you have in the imaging might increase. And how easily is this possible?
So, yeah, in our case, we actually see opposite, that increasing temperature actually increases contrast, and that's what we see here, is that basically more light you put, your more temperature change, and more temperature change creates a bigger contrast in terms of this modulated contrast without heating and with heating.
I think the question was more to the ambient temperature, or how the ambient temperature.
Oh, just cool it down?
No, Paul. It was to one part, yeah, with respect to the simulations, but additionally, there are of course also specific bands which vary a lot with temperature, yeah, especially bands which you have in connection with H bonds. Mm-hmm. And my question is, yeah, how easily is it possible to vary the temperature with your experimental setup?
Yeah, this is a very nice question. Yeah, so yes, you can have some spectral wandering of some bonds with temperature, and to vary temperature, it's again going to depend on particular sample of interest, and like its thickness, its size, because we can estimate back of the envelope temperature changes, and we can vary incident power intensity very accurately, and we can again make a calibration curve of signal and estimate what the temperature change gonna be there, and very accurately change temperature. But for samples that are not heterogeneous, they should be very similar in size to accurately tell that yes, this temperature change is 1 Kelvin, or this is 10 Kelvin. Is this does this make sense for?
Okay.
Still thinking about that.
For the moment, yeah. Thank you.
Okay. I have another question from the chat from Mats Liebe, which is also actually my question. You showed just a simulation before where you have refractive index changes on the order of 10 or 7 times 10 to the minus 3, while the thermal refractive coefficients are typically on the order of 10 to the minus 4 or 10 to the minus 5. So which kind of temperatures do you assume to have in the experiment, or temperature influence?
Yeah, so for these simulations were done by Greg Hartland, but here all temperature jumps are under 100 kelvins. So it's below temperature glass transition for this material. So this is polymer, right? So if you heat it too much, it will transform into glass. So it should be below it, and yeah, under 100 always.
Okay. Do you have any kind of experimental evidence for the temperature in the polystyrene particle?
So sometimes I can melt them, and I can say that yes, this was above this temperature change was above melting point or above glass transition. But other than that, yeah, I didn't measure it. But as I mentioned before, I can accurately trace changes to the particle field, and like when we hit glass transition, for example, I will know this final point, and I will know starting point, and like I can create a calibration, accurately predict measurements. But other than that, we didn't do precise measurement of temperature.
Okay. So other questions for Ilja?
Maybe I will make a quick question.
Yes.
So in the size dependence, where you varied size from 100 to 500 nanometers, and you have a two effect, one effect by the scattered by the particle itself, and then you have a thermal lens effect. And so I will assume that if you increase the particle size, then the scattering effect should increase with the particle size. So I don't see really, in your simulation that it looks like more or less independent of the particle size, the scattering contribution.
Yeah, this is actually a very nice question and quite complicated. And like if you look into theories, there's a lot of thermal lensing effect and so on. But difference there is that you have dipole that is absent in absence of heating, and then you have a big thermal lens. Here's something else happening. You always have a particle that is always scattering light, and then you have a small thermal expansion. And just to put you into context how small the thermal expansion is, like changing 10 kelvins for 1 micrometer particles will change size just by 1 angstrom. So thermal lens effect is always very small there. So it's all scattering. Does it make sense?
Okay, yeah, thanks.
So may I ask maybe a last question? So in the images of the individual polystyrene particles, it looks from what you showed and also in the literature always that there's a kind of halo around the particle. So so yeah, you see here on the right side for the 150 nanometers. And my question is, where is this actually coming from? If it's not in a medium and it's just on a glass slide, is that from the substrate?
Yeah. So, or this is again a very nice question and right in the middle. But so probably this is just a so-called mirage effect when you have heating of air that it refract light a little bit. Okay. Or it's maybe just airy disk of my beam. Maybe I just a little out of focus. This sounds bad, but I assume it's small contribution from air that it's, if particle is heat to, let's say, 50 kelvins above the room temperature, it's going to change refractive index just significantly enough to check, to measure it. I usually always surprised to see them because thermal optic coefficients of air is 3 orders of magnitude smaller than polymer. So this is
okay.
This is a lingering question. This is a
okay.
Nice.
Okay, so there's some comment on that in the chat. Light deflection is one of the effects probed by in PHS. This is actually a question. Should you take into account the distance between the sample and the detector?
Oh, okay, this is a question I know it's sort of, but usually we detect collimated light, so we collimate, so we collect emission, collimate it, and then trace it to detector in a collimated version, then focus on detector. So I assume there is no difference between that, but experimental evidence or experimental measurement of this I never performed. So like short answer to a question, I don't see a reason why it would happen in our geometry when we use collimated light. Okay. But physics, not sure. But thank you. Yeah.